If it doesn't have that gene, which most bacteria don't have that gene, what do you think is going to happen when you put ampicillin on it? They die. So if we go back to our bacteria here, and this one has this plasmid, it can make this resistance gene. We add ampicillin. Does it die? No, it survives. These guys did not get plasmid. We add ampicillin, they do die. So all the plates that I'm going to give you today already have ampicillin in them. So you don't have to add it. You can't really tell it's there, but it is. So all these guys already have ampicillin in them. So when you add your bacteria that don't have anything, you're all just going to kill them. And hopefully some of them will get your plasmid, and they will form those colonies that you saw earlier. Any questions there? Yeah?
Wait, so ampicillin is a chemical or is it another bacteria? So it's a chemical?
It's a chemical. Ampicillin is, it's like, to be fair, we use it as a medicine. It is...
It's in a lot of things.
It's in a lot of things, yeah. There's, to be fair, we use ampicillin resistance for a lot of things. But yeah, it's just a chemical. So it is, any of these antibiotics, for the most part, we grow up, well, we've synthesized a lot of them, and then we'll use them in biology or in medicine or in these kinds of things. So anytime you have an antibiotic, it's some level of chemical. Any other questions? That's a great question, by the way. Cool. All right, so we are at a point where a lot can go wrong. You have a tube that has clear liquid in it that should or should not have done something. It's very hard to know what went wrong or where something went wrong along the way. Things that can go wrong is that maybe that ligation didn't work. Maybe those mutated points didn't get stuck back together. So now we don't have a circle. Are you going to be able to express your protein if it's not a circle? No. Those cells are going to die. You aren't going to be able to express anything, your antibiotic resistance or your GFP. Those cells die. Maybe you didn't get rid of that original template, so you're going to have some of that GFP floating around. Maybe your mutations didn't work and I gave you bad primers, and instead of turning something blue, I turned it to nothing color. These are all possibilities of things that could have happened. Mutations didn't work, PCRs didn't work. We just can't tell until we do the next step. Other possibilities, we live in a bacterial world. There's bacteria on you, there's bacteria on that desk, there's bacteria on pretty much everything. Hopefully not what I'm giving you as reagents, but it's possible that there is. So you can have contaminating DNA in your reaction as well. That's not great. And then there's also a possibility that your transformation just doesn't work. So when we do these, you've got to have controls. You're not just going to transform in your mutated plasmid. Because if you do, and you get something that's not exactly what you expect, it makes it very hard to analyze and figure out where you went wrong. So we're going to have two controls. One is going to be a positive control. So this has two jobs. So we're going to transform cells with something that we know should work. I have never tried a transformation with pure plasmid, which is what I'm going to give you, that has not given me any colonies. So if you are able to transform cells effectively, you should get colonies. So this will tell you, one, that your transformation procedure worked. It is possible to mess up a transformation. Totally possible. So if there are no colonies, that's a pretty good indication that your transformation did not work. That can also mean, oops, if your transformation didn't work for something that's really good, and then you get no colonies on your mutated plate, it probably just means that your transformation didn't work at all. So maybe you did make your mutation, you just didn't get colonies. So this is a good control for figuring out whether this step today worked. It's also a secondarily good control because you are going to transform with your original GFP. So it's going to glow your original green, what you started with. You guys are mutating to YFP yellow, BFP blue, and CFP cyan. Those aren't that far off from green. So it's going to be really nice that you guys are going to be able to look at the color of your new bacteria and compare it to the original GFP or green bacteria. So bonus, two controls. You guys are going to use this, hopefully. Next class period, you guys do this. Secondarily, we're going to have a negative control. You guys are going to transform, but not add any DNA. So you're going to do exactly the same thing that you're doing with all of your transformations, but no DNA. And then you're going to plate that out onto a plate with antibiotic. Should those cells survive? No. They don't have your antibiotic resistance. They should not survive. If they do survive, that means that the rest of your experiment is probably not that great. You probably have some sort of contamination. It may mean you have contamination all across your experiments. So this is a great control for how clean you are, how good you are at doing these procedures in a sterile manner. Because if you're not able to get something, ampicillin, to kill off things that don't have your plasmid, it's not a great sign. And then the last thing you're going to transform is your actual mutated DNA that will hopefully do something different from either of these and be a beautiful color. Fingers crossed. So, the top one, the first one has just your bacteria that has been transformed with pure plasmid that is GFP. So you see green colonies. Your second control is your negative control that you didn't transform with anything. It should have no colonies. Perfect. Underneath that, you have some scenarios of what might happen. So we're going to talk a little bit about what I'm expecting, and then you guys are going to fill this out, and we're going to talk about it hopefully by the end of class. So this first one, this is what your plate, your experimental plate, possibly could look like. So assume everybody had primers for blue fluorescent protein. You have only blue colonies. What is your conclusion that you can draw from this experimental outcome? Everything went right. So yes, that is a good conclusion, but I want you to talk a little bit more about what happened on a molecular level. So yes, everything went right. You were able to mutate your DNA, right? You changed the nucleotides in the GFP sequence to the nucleotide for the BFP sequence. So be specific when you say something, hey, something worked. What do you mean by that on a scientific level? And in some of the later ones, you have less successful transformation. So if they didn't work, what does that mean? Yeah.
So in this instance, wouldn't it mean that we were successful in opening the DNA sequences and inputting an enzyme?
You guys were successful in mutating, for BFP it was just one nucleotide that you were changing. So you were successful in mutating that one nucleotide to the specific nucleotide that you wanted to, that changed the sequence of GFP to BFP. So that's the kind of conclusion that I'm looking for. Now, in some of these later scenarios, there may be more than one possible outcome. Write down as many as you can think of. So there's not just one right answer for a couple of these guys. Also, just so you guys know, in that third one where it has black colonies, they're not black, they're just not colored. They have no fluorescent protein in them, so they're just uncolored colonies. All right, go ahead and take about 10 minutes to try to finish filling this out. You can talk with your groups. It doesn't have to be a solo endeavor, but try to get conclusions for what each of those possibilities would be. So in that next one, it doesn't look the same as this one that we thought was a success, but why is that possible? What could have happened there? And there are at least 2 answers for that one, I'll tell you that.